Chapter 5. Tools of Genetic Engineering 3 Cloning Vectors. Similar to DNA enzymes, the other most important requirements for recombinant DNA technology is the cloning and expression vectors. The recombinant DNA is produced by cloning a foreign DNA isolated either from the genome or synthesized chemically or as cDNA using mRNA molecule. However, cloning of this DNA can be done only when another DNA molecule is available that may replicate in the transformed host cell. This other DNA molecule, used for joining the foreign DNA, is called vectors. The vectors are the DNA molecules that can carry a foreign DNA segment and replicate inside the host cell. Vectors may be plasmids, a bacteriophage, cosmids, phagemids, transposons, a virus YAC or BAC. Moreover, a vector must possess the following characteristics. 1. It must replicate, through origin, in host cells after its introduction. 2. It must contain marker genes such as teta, for tetracycline resistance CANA, for kanamycin resistance AMPR, for ampicillin resistance. These help in selection of transformed cells from untransformed cells. 3. A unique cleavage site must be present in one of the marker genes. By inactivating it, through restriction enzyme 1 can detect the recombinant molecules. However, many vectors used commonly contain several recognition and cleavage sites for several restriction enzymes in a small region. These are called multiple cloning sites, MCS, or polylinkers. Presence of MCS facilitates the use of restriction enzymes of choice. 4. It should contain specific control systems like promoters, terminators, ribosome binding sites, etc. so that the cloned DNA should express properly. The best known of these vectors is the plasmid vector. The largest number exists for E. coli. During the early 1970s, Stanley Cohen and Herb Boyer selected E. coli strain Ki2 for conducting research work on molecular biology. Since then, E. coli K12 has been used for various experiments in recombinant DNA technology and production of products. One of the unique features of E. coli is that it is found in human intestine and cannot thrive outside the laboratory environment. E. coli strain K12 mutant in restriction modification system is used in gene cloning. E. coli strain Ki2 mutant in N gene encodes DNA-specific endonuclease, increases the yield of plasmid DNA and improves DNA quality. The RECA gene is associated with expression of recombination proteins. Therefore, E. coli strain Ki2 mutant in RECA gene ensures about the biosafety of E. coli strain K12 because they can easily be killed by ultraviolet light. A bacterial plasmid vector. The size of plasmids varies from I to 200 kilobits. It depends on host proteins for replication, maintenance and other functions. They are present in a characteristic number of copies in a bacterial cell, yeast cell, in eukaryotic organelles such as mitochondria. The plasmids are maintained as single copy per cell and they are called single copy plasmid. There are multicopy plasmid also which are maintained as 10 to 20 copies per cell. Apart from this, when the plasmids are under the relaxed replication control, they get accumulated in the cell in very large copies, i.e. over 1000 copies per cell. Due to increased yield potential, such plasmids are used as cloning vectors. The colal plasmid replication system of E. coli is most extensively studied. It expresses colicins, which is an antibacterial protein. The colal transcribes a DNA sequence near the ORI gene. Due to increased yield potential, these plasmids are used as cloning vectors. Several plasmids consist of a replicon, a segment of genome that contains an origin of replication, of PMB1, originally isolated from its close relative, colal replicon. In each bacterial cell about 25 to 20 plasmids are maintained under normal growth conditions. The colal or PMBL replicon relies mainly on long-lived enzyme from host. Replication is primed by an RNA primer and takes place unidirectionally from a specific origin. E. coli also consists of another plasmid called fertility factor or F plasmid, which is associated with conjugation. It is about 95 kilobits long plasmid and transfer F factor to FF deficient cells. One copy of F plasmid is present in each cell and encodes about 100 genes. Some of them are needed during the transfer of F factor. F plasmid forms sex pili, the number of which is governed by the of plasmids. It also encodes cell surface binding proteins for the filamentous bacteriophage M13. Plasmids are the extrachromosomal, self-replicating, and double-stranded closed and circular DNA molecules present in the bacterial cell. Plasmids contain sufficient genetic information for their own replication. A number of host properties are specified by plasmids, such as antibiotic and heavy metal resistance, nitrogen fixation, pollutant degradation, bacteriocin and toxin production, chelicin factors and phages. Naturally occurring plasmids can be modified by in vitro techniques. N. Cohen and co-workers in 1973 for the first time reported the cloning DNA by using plasmid as vector. A plasmid can be considered a suitable cloning vehicle if it possesses the following features. 1. It can be really isolated from the cells. 2. It possesses a single restriction site for one or more restriction enzymes. 3. Insertion of a linear molecule at one of these sites does not alter its replication properties. 4. It can be reintroduced into a bacterial cell, and cells carrying the plasmid with or without the insert can be selected or identified. Bernard and Helensky, 1980. 5. They do not occur free in nature but are found in bacterial cells. In many cases, the principal objective of cloning experiment is the insertion of a particular restriction fragment into a suitable plasmid vector and its amplification. Amplification of plasmids. Amplification is a process of increasing the number of a plasmid in a bacterial cell. In this process, a cell containing a relaxed plasmid is treated with a drug to inhibit protein synthesis. Consequently, cells stop replicating. The relaxed plasmid pBR322 continues to replicate despite of drug treatment. Replication of relaxed plasmid neither depends on cell replication nor requires protein synthesis. For example, addition of chloramphenicol causes pBR322 to get increased from 3 to 5 to about 3000 per cell. Finally, the ratio of plasmid DNA to chromosomal DNA is increased, which makes easy to isolate the plasmid DNA. In order to replicate and clone a fragment of foreign DNA, it is necessary to possess a sequence of nucleotides which are recognized by the host bacterium as an origin of replication. The origin of replication occurs naturally in plasmid and transferred to onward progenies. During cell division, these genes are transferred, resulting in gradual decrease in number of such genes. However, due to continuous exchange of genetic materials between plasmid and chromosomal DNA, new genes originate with respect to environmental conditions. There are several plasmid cloning vectors such as pBR320, PSC102, CountyIL, PUC, PRP4, PUC2, PRSF-LOLO, PWWO, T and re-DNA plasmids, etc. The plasmid cloning vectors are designated by P, some abbreviations and a few numbers. For example, in pBR320, P means plasmid, BR refers to the researchers, F. Bolivar and R. Rodriguez, who discovered the plasmid, and 322 is the numerical designation. Some of the plasmids are given in Table 5. 1. pBR322. The plasmids that occur naturally do not possess all the characteristics to be used as cloning vector. Therefore, they are constructed by inserting the genes of relaxed replication and genes for antibiotic resistance. The pBR322 is the first artificial cloning vector developed in 1977 by Bolivar and Rodriguez from E. coli plasmid coli. It is 4.362 kilobits long and most widely used cloning vector. In the transformed E. coli cells, its copy number remains about 15 to 20 molecules per cell. However, its number may be amplified by incubating the transformed E. coli cells on a medium containing chloramphenicol. See Table 5.1. Some cloning vectors. Asterisk. IKB kilobase pairs equals 1000 base pairs equals 0.66 megadalton. Asterisk. Resistance to ampicillin, AMP, tetracycline, TET, erythromycin, M, streptomycin, STRR, kanamycin, CANT, rifampicin, RIF, and calicin production, LIMM. A physical map of plasmid pBR322 is shown in Figure 5.1. The pBR322 is constructed from the plasmids of E. coli, pBR318 and pBR320. It contains origin of replication, ORI, that was derived from a plasmid related to naturally occurring plasmid coli. Therefore, its replication may be more faster than bacterial DNA. It also possesses genes conferring resistance to antibiotics, e.g. ampicillin, AM, and tetracycline, TETA, and unique recognition sites for 20 restriction endonucleases. 6 of these 20 sites, such as ECARIV, BAMHL, SPHL, SAL, XMAL and NRUL, are present within the gene coding for tetracycline resistance. 2 sites, HINDL and CLAL, are located within the promoter of the tetracycline resistance gene, and the 3 sites PSTL, PVUL and SCAL, within the beta-lactamase gene that provide resistance to ampicillin. If any foreign DNA is cloned into any of these 11 sites, insertional inactivation of any of the antibiotic resistance markers takes place. For example, if a foreign DNA molecule is inserted at TETA gene cluster, the property of tetracycline resistance will be lost. The recombinant plasmid will allow the cells to grow only in the presence of ampicillin but will not protect them against tetracycline. The presence of an antibiotic-resistant gene in a plasmid of bacterium will confer resistance to that antibiotic. The antibiotic-resistant cells can be selected by culturing the cells on nutrient medium supplemented with ampicillin or tetracycline. Figure 5.1. A map OF pBR322, 4.36 kilobits, showing a number of restriction sites and regions encoding for resistance to ampicillin, AMP, and tetracycline, TETA, and origin of replication, ORI. 2. PUC vectors. In 1983, Messings and co-workers developed the PUC vectors at the University of California. These plasmids consist of coli ORI, origin of replication, gene, AMPR gene, for ampicillin resistance, latch, gene, the shortest derivative of latch operon for E. coli SS lactamase. The PUC19I, one of the popular plasmid vectors of PUC family, Figure 5.2. The engineered version, latch, of its latch gene consists of multiple restriction sites within the first part of the coding region of the gene. This region is known as multiple cloning site, MCS. If a target DNA is inserted at any of these sites, the latch gene is inactivated. Hence, such recombinant vector will form white colony on a suitable plate as compared to non-recombinant vector. In cloning experiment, the choice for application of restriction enzymes is increased after using as MCS. A MCS at the 5 end of latch is given below. See the figure. The PUC series consist of several plasmids such as PUC8, PUC9, PUC12, PUC13, PUC18, PUC19, etc. The PUC19 is called as general-purpose plasmid cloning vector. Such vectors are designated for cloning of relatively small, less than 10 kilobits, DNA fragments, often in E. coli at MCS. It consists of 2,686 base pairs and possesses an AMPR gene, latch gene, for SS galactosidase, and LACL gene, for production of repressor protein to regulate latch. In PUC19, MCS is incorporated into latch gene without interfering the function of other genes. The MCS has the unique sites for several restriction endonucleases such as ECRI, SACL, KPNL, XMAL, SMAL, BAMHL, XBA1, SALE, HINDL, ACCL, BSPMI, PSTL, HINDL. It also carries one ORI gene from pBR322. When the cells carrying unchanged PUC19 are grown in the presence of isopropylthiogalactosid, ITCO, an inducer of lac operon, the production of LACL gene cannot bind to the promoter operator region of the latch gene. Therefore, the latch gene encoded in the plasmid is transcribed. The LAC-Z protein combines with chromosomal DNA transcribed protein and forms an hybrid SS galactosidase. The MCS is tagged in the latch gene of PUC19 plasmid, which does not interfere the production of hybrid SS-galactosidase. On the other hand, if a substrate 5-bromo-4-chlorionolyl SS-galactosidase X equals gal is present in the medium, the hybrid SS-galactosidase hydrolyzes it to produce a blue product. Therefore, colonies containing unmodified PUC19 appear blue in color. When the DNA insert is cloned in latch gene, this gene will be inactivated and will not express SS-galactosidase. Consequently, the colonies formed by E. coli cells containing recombinant plasmid will be white in color. This plasmid vector is designed for blue-white screening on X-gal substrate. 3. PASIC 84. The plasmid PASIC 184 was developed by using P15A plasmid. It is a small-sized plasmid, 4 kilobits. As described in pBR322, it consists of several restriction sites, CHLR, for chloramphenicol resistance, and TETA, for tetracycline resistance genes. The plasmid containing cloned DNA fragments can be detected by insertional inactivation of one of the antibiotic resistance genes. Its number can be amplified, unlike pBR322, as the latter is not self-transmissible. Fig. 5.3. 4. Agrobacterium-based plasmid vectors. Several groups of plants have received attention as a means toward DNA cloning and expression of foreign DNA in plant cells. The DNA viruses of plants are of two types. 1. Double-stranded DNA viruses, as cholamoviruses, and 2. Single-stranded DNA virus, for example Geminivirus. Although plant viruses have major scope for the development as cloning vector, yet this aspect has not been studied so far. A good deal of work is done on soil-borne bacteria, Agrobacterium tumefaciens causing crown gall disease and a rhizogenes causing hairy root disease on the stems of numerous plants. A. T. plasmid. A tumefaciens consists of a large plasmid which induces tumor in plants, therefore, the plasmid is called as T. plasmid. The size of T. plasmid ranges between 180 and 250 kilobits. It contains tDNA region of about 23 to 25 kilobits, which is transferred into plant cells. T. plasmid can be grouped into 3 on the basis of opine types, for example, octopine, nopoline and agropine. Structural formulae and diagrams of these are given in Figs. 5.4 and 5.5, respectively. Left side of tDNA is known as tltDNA and right side of the same as trtDNA. Both the essential tDNA and non-essential regions of T plasmids, both octopine and nopoline types, contain many genes. One or more unusual amino acid derivatives encoded by DNA is known as opines. Opines are neither found normally in plants nor required by plants. See the figure. Figure 5.4. A map of octopine plasmid, A, and nopoline plasmid, B. A tumefaciens uses it as a source of carbon and nitrogen for the growth and multiplication. The genes responsible for the biosynthesis, OCS gene for octopine and NOS for nopoline, and degradation, OCC for octopine and NOC for nopoline catabolism, of opines are located at the well-defined regions. ARC genes are required for arginine catabolism. The plasmid also carries transfer, TRA, gene to help the transfer tDNA from one bacterium to other bacterial or plant cells, ONC gene for oncogenicity, ORI gene for origin of replication, and INC gene for incompatibility. See the figure. Figure 5.5. Structural formulae of octopines, A, and nopolines, B. I. Mechanism of DNA transfer. A tumefaciens lives in soil and attack many dicotyledonous plants, figure 5.6a, most probably at the level of soil surface. Formation of wound is necessary for establishment of bacterial plasmid into plant cells. Lipopolysaccharide, a compound secreted by bacterial cell wall, helps in its attachment with polygolacturonic acid fractions of plant cell wall. From the wounded cell walls of plants, a phenolic compound of low molecular weight, acetosyringone, is secreted, which induces the Ver genes of T plasmids. Ver genes encode an enzyme which nick the double-stranded tDNA on the same strand at 2 points, and produces single-stranded DNA molecules. A cut is made on right border of tDNA and a single-stranded toner fragment of 5 feet 93 direction is generated. It is carried into plant cells b. Figure 5.6. Process of infection by Agrobacterium tumefaciens on a dicot plant and induction of tumor or gall AD, TP, tDNA of plasmid, PN, plant nucleus, BC, bacterial chromosome. tDNA of T plasmid is stably integrated with plant DNA. The sequencing of nucleic acid at the junction between plant DNA and tDNA has been done. T region in both octopine and nopoline plasmids is flanked by a direct repeat of 25 base pairs. OPS gene encodes enzymes for the synthesis of apines in transformed cells which are required for proliferation of infecting bacteria. TLT-DNA encodes 2 enzymes that are involved in biosynthesis of 2 phytohormones, auxin and cytokinin C. This results in disorganized proliferation of shells commonly known as callus, gall or tumor D. Therefore, the galls are colonized by the bacteria. 2. Tumor morphology. Several gene loci for controlling the morphology of tumor have been identified on tLDNA of 2 octopine plasmids. These loci are grouped into 3 categories: TML, which causes larger tumor, TMR, which induce tumor with large number of roots, and TMS, which causes tumor with large number of shoots, figure 5.7. These effects are due to the products of loci of tLDNA on the cytokinin-auxin ratios within the transformed tissues. The function of TML is not known so far. Figure 5.7. Genetic map of octopine tLDNA of T plasmid controlling tumor morphology. B. Re plasmid. Agrobacterium rhizogenes induces adventitious roots in plants. This rhizogenicity has been correlated with the presence of a large plasmid, the Re plasmid, for root induction parallel to T plasmid. A rhizogenes does not induce enzymes for the biosynthesis of auxin or cytokinin. Plasmid for this species also contains atDNA which causes the development of hairy roots. It has also been reported to confer drought tolerance on apple seedlings. The possible advantage of using Re plasmid of a rhizogenes is the production of secondary root systems for 1. Better anchorage of plants, 2. Ability to resist anoxia from flooding of the soil, 3. Increased drought resistance due to high root density, and 4. Greater chance of interaction with soil-borne mycorrhizal fungi. It may be applied to establish the secondary root systems in certain plants, fruit trees, by inoculation of soil, as well as be beneficial for those plants which are propagated through cutting and rooting as stimulants for root formation on cuttings. C. Properties of T and Rhi-based plasmids. Agrobacterium plasmids have been used for introduction of genes of desirable traits into plants. The properties of T and Rhi-based plasmids are as below. 1. They do not cause disease because of being disarmed by removing disease-causing gene. 2. They possess sites for insertion of foreign gene which needs to be introduced. 3. They possess selectable markers, i.e., genes which help in selecting the transformed cells. The selectable marker is a powerful promoter, e.g. 35S, for high level of expression of derived from cauliflower mosaic virus (CAMF), and polyadenylation signal for adding mRNA. The most commonly used selectable marker is neomycin phosphotransferase (NPTLL), that confers resistance to neomycin and allows the cells to grow in medium containing neomycin. Thus, after inserting a foreign gene, a vector is designed. The vector is transferred to a tumefaciens or a rhizogenes depending on cases. The transformed bacterium is used to infect the designed host cell where the transformation of gene is to be brought about. For gene transfer, generally explants are used. The explants are protoplasts, suspension of cultured cells, clumps of callus, thin cell layers, epidermis, leaf discs, sliced tissue, floral tissue, stem and root selection. The explants are incubated or co-cultured with agrobacterium containing vector with modified foreign gene. The transformed cell or colonies are selected due to the selectable marker and used for regeneration of whole plant, which are expressed to be transgenic in nature. These regenerated plants can again be tested for the transfer of gene with the help of several scoreable or screenable markers called reporter genes, which is also transferred along with desired genes. The most commonly used reporter genes are NPTLL, CAT, GUS and LUX. For details see Chapter 13, Transgenic Plants. In addition to E. coli, some of the gram-negative and gram-positive bacteria have also investigated and used as cloning vector for medical and agricultural studies. Recently, a broad host range cloning vehicle has been developed from RK2, which is a plasmid of PL group of gram-negative bacteria. RK2 plasmid contains a single restriction site for ACOR1, HIND3, BAMH1 and BG3. However, a broad host range cloning system has been developed from it by incorporating the transfer and replication regions of this plasmid into two different plasmids. The three regions of RK2 plasmid essential for replication, e.g. ARIV, TRFA and TRFB, have been incorporated into the plasmid cloning vector, the PUC 290. B. Bacteriophage vectors.